everyone. Welcome to episode 95 of Cell and Gene, the podcast. I'm Erin Harris. Thanks for tuning in. On this episode, I chat with Helen Sabzevari, Ph.D., President and CEO of Precigen, a discovery and clinical stage biopharmaceutical company based in Germantown, Maryland, that specializes in developing treatments for immuno-oncology, autoimmune disorders, and infectious diseases. Dr. Sabzevari discusses Precigen's groundbreaking work, particularly their recent BLA submission for PRGN-2012, a potential first FDA-approved treatment for adults with recurrent respiratory papillomatosis, or RRP. She also breaks down the key advantages of using gorilla adenovectors for delivering large genetic payloads, and a whole lot more. I was so excited to have Helen on the podcast. I met her in person 2 meetings on the Mesas ago, and I'm so thrilled to have her on the podcast finally. It's been a long time coming. You'll take a great deal of actionable information away from this episode, so let me get out of your way so you can tune in. Here's my conversation with Precigen's Dr. Helen Sabzevari. Enjoy. Helen, hi. Welcome to Cell and Gene, the podcast. Thanks for your time.
Thank you, Erin. It's great to be here, and thank you for the invitation.
Absolutely. I feel like it's long overdue. I know we see each other quite a bit in industry events, and so I'm glad to finally have you on the podcast. It's, like I said, it's long overdue. So talk to us a little bit about Precigen and, what especially the company's working on in 2025.
Absolutely. So Precigen is a cell and gene therapy company with two major platforms that we have. It's our well-differentiated gorilla adenovirus platform and also our truly overnight autologous ultra CAR T platform. And what we are extremely currently excited about both platform, but what is the highest priority and moving very rapidly, it's about our gorilla adenovirus platform and program PRGN-2012, which is in a recurrent respiratory papillomatosis. And we just submitted our BLA. So we are very excited for our patients, for the company, and really changing the company, going from a R&D toward the commercial company.
Excellent. So you mentioned, of course, your gorilla adenovectors. That's what I want to start first. Talk to us about how they, in Precigen's adenovirus platform, differ from, say, traditional adenovectors in terms of immune response and repeat administration potential.
Absolutely. So as our audience are aware that the adenovectors in general have been used over the past decades and successfully for various type of prophylactic vaccination in different indication. But one of the major limitations that the classical adenoviruses have, and also it applies to all the other ones as well, similarly, is the limited amount of time that you can use it. It's not that they have not been used in a repeated fashion. They have been used, but the problem is that their efficacy becomes limited after one or two, usage. And the reason is that you have these neutralizing antibodies that generally get produced from patients, and that, by the name itself, it neutralizes the efficacy of these vectors and the ability of the T cell to basically do the work. How gorilla adenoviruses are different, and by the way, we have the full IP around this platform, and it's very unique and differentiated, is that not only you can give this number of times, but upon giving them in a repeated fashion, you keep enhancing the specific CD8 T cell responses. And right there, it's completely differentiated from all the other viruses and all the other classical adenoviruses. And we have shown that in a number of clinical trials that you can go to upward of 16, 17 times, giving it on a monthly basis, and you keep enhancing your specific CD8 responses. The other part that our gorilla adenovectors are differentiated from the rest in the field is the capacity. When you look at the other adenoviruses, the top capacity is around 5 kb. So the number of genes or epitopes that you can fit in is limited. And with that limitation, therefore, evoking the immune systems, you're going to be generating certain number of, let's say, specific T cells only to certain epitopes. Gorilla adenovectors, they have a 12 kb capacity. What does that mean? That means you can put much more epitopes, much more genes, and also you can basically be very innovative in the design of the vector and the way these epitopes are linked together or the genes are put together and controlled. And as a result of that, you can have a much more comprehensive CD8 response that is not just to one clone or to one epitope, but it can be multi-epitope that covers the indication or a specificity that you need if you are battling an infectious disease or if you're battling a cancer in that sense. Because as we know, one clone is not necessarily always would be enough to deal with that. And I think in that level, also it's completely differentiated. And by the way, we have done all of the preliminary data in healthy donors, and we have looked at the seropositivity or a pre-immunity to these vectors in thousands of healthy donors, both in the United States and also across Africa. And either individual have no pre-immunity or seropositivity at all, or very, very little. And that is the reason that when you keep it, giving it multiple times, you are not generating the same extent of neutralizing antibodies. And also one other specific thing about these vectors, it pushes more toward the CD8 responses as opposed to humoral responses. And I think that's the major differentiating factors here.
Good. Thank you. Okay. Taking it a little bit down a different path, what would you say are the advantages of using gorilla adenovectors, for delivering large genetic payloads, and how does this impact, say, the development of therapies for complex diseases?
I think that's a great question, Erin. Well, if we look at, for instance, our PRG in 2012, which is in a rare disease for a recurrent respiratory papillomatosis, RRP. Now, the root cause of this disease is HPV 6 and 11, and that leads to basically having these benign tumors reoccurring continuously, either in the trachea or on the vocal cords of these patients. Now, when you look at this and we say, okay, HPV 6 and HPV 11, there are not just one epitope here. First of all, you have number of genes, you have number of epitopes, and it becomes very important. How many of these epitopes can you target? And classically, people have been looking at the known epitopes for HPV 6 and 11, and because of a limitation that it's there, you can only put certain amount of these epitopes in there. What we have done, for instance, in our PRG in 2012, because we have a 12 kb capacity with our gorilla adenoviruses, now we have not only put all the classical epitopes that we have, but we have identified other epitopes, which are we have a full IP around. And we have designed a specific way in the way these are linked together that has a much higher coverage. And therefore, you can generate, for the lack of better word, an army of T cells that will see majority of these epitopes that may exist in these benign tumors in the patients. And when they see that, then they are capable of destroying. Whereas when you have a limitation in a number of epitopes, your army is only qualified to see a certain part, and they can destroy only a certain enemy. However, the army that is broader and can see more epitopes, it can destroy a much further ground in the sense of these infected cells and benign tumor cells that they cause this basically repeated growth on the trachea or in the vocal cord. And that is one of the major differences. And then, obviously, based on that capacity and based on the ability that you can repeat those for a number of times and keep enhancing and training and activating this army further and further of this specific CD8s, now you have a much broader reach in this indication as far as treatment is concerned.
Sure. Okay, thank you. All right, so can you then elaborate on the potential applications of the Adenovirus platform beyond recurrent respiratory papillomatosis, or RRP, particularly in other HPV-driven indications, perhaps such as cervical cancer?
Yeah, again, wonderful question. I think RRP, of course, we started this as a rare disease, and it was because for the past more than half a century, there has been no treatment for this disease at all. Many things have been tried. Everything practically has been tried, but really there's no approved treatment for this. And we started there. But other indications, even indications that are caused by HPV 6 and 11, such as genital warts, which millions of people in the United States and tens of millions, if not hundreds of millions, outside of the United States are affected by this infection, and it's also very devastating. It can be now the similar treatment with our gorilla adenovirus can be applied for that indication, or in the cases of throat cancer. On the other side, the exact same backbone that we have used for our Precigen 2012, which is this gorilla adenovectors, we have used to address HPV 16 and 18, which causes all the HPV-related cancers, as you mentioned, such as cervical cancers, head and neck, and anal cancers, which is 5% of all cancers occurring. So hundreds of thousands of patients per year, basically they develop either cervical cancer or head and neck cancers, anal cancers. It's a huge indication with unmet needs. And what we have done, our Precigen 2009 have been designed using the same gorilla adenovectors to address HPV 16, 18. And we actually reported at ASCO for phase 1 data that we showed. And it was very exciting because we not only show that in a stage 4 patients that they have relapse and they are refractory to even checkpoint inhibitors in cervical cancer patients or head and neck, that when we administered the Precigen 2009, not only it has a very favorable safety, but also we showed 30% objective responses. These patients that they have failed everything else and there was nothing in front of them. Now we had these patients, some of them achieving complete responses, partial responses, and a durability of the responses were quite high as well. So we are really excited about this platform and the ability that it has from a gene therapy perspective to, from within, activate your own specific CD8 responses that can now battle the various cancers such as HPV. And we think that these have a huge potential on its own and in combination with other therapies that can really advance the field. And if you look at the response rates, for instance, in HPV-related cancers, with checkpoint inhibitor, which is the best tool that we have in our hand at this moment, cervical cancer, 15% response rate. Head and neck, 18% response rate. When you are looking at now the Precigen 2009, based on the phase 1 data, and we have taken this now to phase 2 data, both in head and neck as well as cervical cancer. Now, in patients that actually have failed checkpoint inhibitor, we have achieved, based on the phase 1 data at ASCO that was reported, 30% objective responses. So this is already a huge advancement. And we are really looking forward to advancing these phase 2s and the readout for this.
Absolutely. Certainly. I want to talk a little bit about Precigen's ultravector technology and specifically how it impacts, say, the scalability and cost effectiveness of, bringing this into the market. How does the technology contribute to high yield manufacturing of adenovirus vectors, and, what impact does that have on scalability and cost effectiveness?
Well, first of all, we have the cell line that we have for the production of our gorilla adenovectors. This is one of the major advantages. It's very highly productive as far as manufacturing is concerned, and the amount of the viral vector that can produce is quite high. So that obviously makes it much easier to produce high amount of these viral vectors with the cell line that have a great production capability. The ability of putting ultravectors that they contain, or they have a large capacity, for instance, 12 kb. If you can imagine, for classical ways, when the capacity is only 5 kb, this is the maximum that you can fit. And if you try to push this any further, for the lack of better word, what will happen is you reduce your manufacturing ability, and then the cost goes higher, because then you have more manufacturing failures, or you have less amount of the virus that you are producing. And then as a result, actually, then the cost of manufacturing goes up. Or if you are trying to cover a number of epitopes, then you have to go to a number of viral vectors and manufacture different viruses with different epitopes, and then perhaps combine as a combination and go in. In the case of the gorilla adenovectors and ultravectors that we have for this, we can design with the capacity of our gorilla adenovectors, is now you have up to 12 kb. So first of all, you can fit all your epitopes and genes that you like in the same vector. That's number one. Number two, you have a cell line that produces these vectors that are extremely effective and produces high yields. And that's very, very important. So that brings down, obviously, the cost of the manufacturing and the time of the manufacturing. And at the same token, what we have done at Precigen is we had established already our GMP facilities, and now the commercial cGMP facility, that it actually produced all of our own vectors directly at our sites. So there is no really requirement for the drug substance, that it's all produced on the site, and allowed us to move very rapidly to the clinic, and also for the commercial production of this material. So that's how I think it has allowed not only the cost of the manufacturing, the ease of the manufacturing, that you can produce high amounts of it. Especially it becomes very, very important when you have to produce large doses for indications such as head and neck or cervical cancers. Obviously, there are millions of patients that eventually would be treated globally. And it is extremely important that you have a high yield manufacturing capabilities.
Yeah, absolutely. And I think, and Precigen has been on that, that's been a goal of yours since the inception of your company. And you've correct me if I'm wrong, that's always been what absolutely what you've been doing this entire time, is that really quick turnaround in manufacturing.
Yeah, you're absolutely correct. This is exactly one of the reasons that if you look at our, basically even the history of development of PRG in 2012 for RRP, We started the discovery in 2020. We went to clinic in 2021. All of this was taking place in the middle of COVID, of course.
Mm-hmm.
And one of the reasons that we were able to keep up the speed and get to the point of BLA submission really from a finishing of phase 1, which was end of 2021 to 2024, in 3 years to submit our BLA for priority review. One of the major aspects has been we have been able to and capable of producing these vectors, adenoviral vectors, directly in our own GMP facility and have it ready for a clinical trial. And there was no delay, even during the COVID time, to get to patients.
Yeah, really, really important and really impressive. And speaking of the BLA submission for PRGN 2012. And so before I let you go, I want to make sure we discuss that in a little bit more detail. What are your expectations for commercial launch? And, how might that, would you say, overall impact the broader field of adenovector-based therapies?
I think, first of all, we were extremely happy when in 2023, based on the data for our safety and efficacy from phase 1, FDA granted us a breakthrough designation and also a prior rapid accelerated path for the submission and reviews. This was really, I think, paradigm shifting for this disease, because up to that point, no therapy has been advanced to that level, and based on the safety and efficacy that we showed, and then we followed at ASCO in 2024, and we showed in another phase, in a phase 2, similarly, we have achieved 51% complete responses in a very severe patient population. And what do we mean by that? Patients that they had required minimum of 3 surgeries in a prior years. And some of the patients that participated in our clinical trials, they had as many as 10 surgery per year. You can imagine, this is a devastating disease. Some of our patients have had upward of 300 surgeries. And because and some of our patients that participated in this trial, they have had this disease from an age of 1 year old. So if you can imagine, even though we were treating adults and adults entered, but they have been basically living with this devastated disease from the age of 1. Can you imagine to take your child on a monthly basis to a surgery? Because there is nothing else. And unlike other surgeries that usually surgeons go in and take out what is wrong, in this setting, the surgery is not curative at all. They know that it's just a matter of, because the patient can't breathe or they cannot talk, and the surgeons, they go in to just clear the airway, for instance, or take the benign tumor from a vocal cord, knowing that these patients come back for the surgery again and again. So based on that data, this was very, very important, and we were very happy in discussions with FDA, and we received the breakthrough designation. And as we just, not only we showed the data at ASCO and we communicated, we have submitted now our rolling BLA by the end of 2024. And actually, as I am speaking to you yesterday, the full result of both our phase 1, phase 2, and we are really excited, came out in Lancet Journal, Respiratory, followed by a commentary by one of the experts, global experts in this field, which they refer to this as a landmark achievement for the treatments of RRP. So you can imagine what that means, and we are really excited about this because, as I mentioned, for more than half of a century, these patients has been living with this devastating disease, and there has been nothing there. And we are really thrilled that we can bring this to the patients. We look forward to, of course, next steps with the FDA review and hopefully receiving the PDUFA date and for commercialization upon approval, of course, of Precigen 2012 as the first FDA approved, if approved, in the second half of 2025 to be commercialized.
So exciting. And certainly we'll be following up with you later this year once that actually does come to fruition, and we can, talk all about the exciting information that's come out since then. But that's so wonderful. Congratulations. I mean,
thank you very much.
You and your team must be really excited and thrilled and kind of, not that you ever want to wish time away, but really happy for the second half of 2025 to arrive.
Thank you. Absolutely. We are thrilled, and we are very excited for our patients, for our organizations, and we are very grateful to all of the patients that they participated and also all of the investigators.
For sure. All right, listeners, that's a wrap for this episode of Cell and Gene, the podcast, featuring Precigen's Dr. Helen Sabzevari. Helen, thank you so much for your time and for the really great information that you shared with us today. And like I said, we'll have to have you on in the second half of 2025, so we'll be back to discuss this more.
Absolutely. I look forward to it, and thank you again for having us.
Absolutely. All right, thanks, listeners, for tuning in, and we'll talk to you next time.